So now we're on the urinary system. This is that chapter that will be also included in our exam 4. So it will go all the way through this chapter. It won't go through the fluid and electrolytes portion. You guys are doing histology evals this week. Don't forget to create your cheat sheets. You can use that. And then also you're doing your renal eval this week. And so the structures that we're going to list today during lecture are going to be structures that you should be able to identify. We won't have a lot of the vascular structures listed today. I'll go over them a little bit, but you should already have those kind of known as you work on then your kidney dissection and that eval today. Kidney anatomy is our first topic here. Retroperitoneal from the 12th thoracic vertebrae to the 3rd lumbar area. One thing I want to actually emphasize to you, the entire urinary system is retroperitoneal. So that means if I take a laparoscope and I look inside your abdomen and look all around, am I going to find anything related to the kidneys? Nothing, because it's all retroperitoneal, so it's outside of the abdominal cavity. And it's, for the most part, it's all posterior until we actually get to the area where the bladder is located, and the bladder is on our pelvic floor, but also when it starts to fill up, fills up on our ventral aspect of our abdomen, but still it's retroperitoneal. If you had a full bladder and I looked inside, I would be able to see there's this thing kind of bulging down into the abdomen. So that's why this is a very different arrangement for the human than what you guys are experiencing with the cats, because those bladders on those cats are right there, aren't they? They're right in your face, they're right up front, easy to see, not that way for the human being. When we take a look at, it always comes up, when we take a look at this picture here, there's structures that you should be able to start to identify. And this would be like a slice of a CT or an MRI of somebody's abdominal area. And so figuring out which is the right side and the left side, I hope is getting a little bit easier for you, because there's some organs that are index organs that are always on the left side or always on the right side. And this organ right here is the spleen. And the spleen is always on the left side. So that's how you can find, if you have an organ that's your index organ, you can find it, and you can start to recognize other things. The liver, remember, it's the largest internal organ that we actually have, and so that's all right here. And the right lobe of the liver is huge and makes up most of the liver, and so you indeed see that liver there on the right side. It's easy to recognize this as the stomach. You can see the rugae, and you know what sphincter this is, right? It's the pyloric sphincter. So which part of the small intestine is this? It's duodenum or duodenum. Either way is perfectly acceptable. And then we see our pancreas right here in the midline behind the stomach, correct? So it's behind the stomach. It's also considered retroperitoneal. And when I go over to this area, I would want to say that's kind of like the splenic flexure of the large intestine, correct? So it's up there by the spleen, splenic flexure leading to the descending colon and that. So that's why as you take a look at some of these pictures to identify very specific organs right now for us, the kidneys, don't forget to take some time to actually orient yourself and make sure you have a good understanding of the regional anatomy. IVC here, abdominal aorta here. Notice that the IVC is a little bit off to the right side. The aorta is a little bit more centrally located in that. And then when we take a look at our left kidney versus our right kidney, why are these sizes different in this cross-sectional view? The right one's lower than the left, and so we're seeing the upper skinnier portion, so to speak, of the kidney, and if we went down, we would then start to see it getting larger in cross-sectional diameter, and then of course it will get skinny again. And so that's a nice thing too. You're taking a look here, realizing that right lobe of the liver is large enough to displace the kidney inferiorly. And so that's why we have this little bit of difference here from the 12th thoracic to the 3rd lumbar vertebrae for that location. So obviously the right kidney's closer to the 3rd, correct? than the left, and the left is going to be closer to the 12th than the right. So we have some of those things just to keep in mind. Going down to the external anatomy here, hilus is the medial indentation. And I want you to realize that hilus is synonymous with hilum. They used to use hilus a long time ago, and now they're using hilum a little bit more. And when you take a look at the picture up above, it says renal hilum. And so realize that is the medial aspect of our kidneys with the way they're actually located in our body, and it's indentation like a kidney bead. And so from there we'll find blood vessels, BVs, plus the ureter enters and exits. Enter would be an artery, exit would be a vein, and ureter would be exiting also. Associated with our kidneys are our adrenal glands. They sit in the fat at the superior pole or portion of each kidney, and because of that, they also have another name called suprarenal. The kidney itself is contained. It has a fibrous capsule, thin, dense connective tissue, and would that be dense regular or dense irregular CT? Dense irregular, because we want to make sure that we can accommodate any irregularities where the kidney might balloon up as if it had a cyst or something along that line. So irregular. Excellent. The kidneys are cushioned in their location. They're cushioned by fat. That's one thing about fat. Fat's cushioning and protecting things and also hiding things from you, I guess you guys have probably already found. But that fat is there to keep them cushioned and buffered, so if you realize there's not a lot holding them in place other than that renal fascia that we're going to talk about in just a little bit. And once you break through all that, then the only thing holding them in their location is the blood vessels, essentially, after you get their dissection done. So realize that perirenal fat, also called perinephric. Nephrons are an important structural and functional unit of the kidney, and so nephron is used a lot because that's actually what makes our filtrate, which eventually will become urine. The renal fascia, going back up here and taking a look at this cartoon drawing, erasing some things so I can see a little bit better around it. That renal fascia is this line right here. So it's kind of keeping that perinephric or perirenal fat in place. So that's an important thing because this is also the main item that actually keeps our kidneys in their location in our abdominal cavity. So that is, again, dense CT. That's going to be dense irregular CT, and it is very important in anchoring our kidneys in their location. We're going to look at the internal anatomy, but we can actually go back to our cartoon and still talk about the anatomy internally from this cartoon itself. What is this area of the kidney called? Cortex. Excellent. And so then the medullary area would then be right adjacent to that. And stop about there. In the medullary area are these guys called pyramids. And then what are the things in between the pyramids called? Columns. So renal columns. And in those renal columns are actually where you're going to find those blood vessels that are called interlobar. So the interlobar blood vessels are in the renal columns. And then arching over the pyramids, the big fat base of the pyramids, which is actually pointing more to the external stuff, is where you'll find the arcuate blood vessels, arching over, arcuate. The other thing too, in your lab news, the blood vessels going off into the cortex are called cortical radiating. They are also listed in other books as interlobular, but we really like the definition cortical radiating because that's where they're located in the cortex and they're radiating straight out from that cortex. So that's a better name because it's more descriptive. And so that's why we actually ask you guys to use those names with that. Going down now to the written notes, let's take a look at the internal anatomy. Again, 3 main regions. I didn't point out the other region here, which is internal to our medullary area, and this is going to be associated with the renal pelvis, and it's got some breakdowns on that. Notes say cortex, light-colored outer region, and indeed it is lighter in coloration, fresh or preserved specimen. Compared to the medullary region, which is inner and indeed darker in color, and the things that give it the darker coloration are the renal pyramids. And it says cone-shaped masses, and like a megaphone cone, we have that inverted look to it, so it's like this. That's a renal pyramid, and this is the area where urine or filtrate is processed. So there's a lot of processing that still occurs in the cortical region, but once we get into the pyramid region, the stuff that will finally come out at the apex of the pyramid is actually urine. And what does that urine drip into? It drips into first the minor calyx. So the minor calyx is designed to be a cup to collect that filtrate, which is now no longer filtrate once it leaves the pyramid. Once it leaves the apex, it is now actually urine. And when we have more than one pyramid dripping urine into its minor calyx, when the two come together, it's where we now have a major calyx. So this here is major, and these guys here are minor. So you have a lot more minor calyxes than you do major calyxes. But remember, these are 3-dimensional structures. A pyramid's a 3-dimensional structure, and so this is like a cup that's reaching up to actually gather the urine that's actually now coming out of that. Renal columns indeed separate the pyramids and contain our interlobar blood vessels. This cartoon to the right is, again, structures that are actually easy to identify: cortex, pyramid, medullary area, columns. Apex. This here is the minor calyx. This here is a major calyx. The funnel-shaped area is the pelvis, and then of course you have your ureter from this area. The outer aspect is the fibrous capsule. This area is called the hilus or hilum, and you'll be able to recognize the renal artery because it's muscular, and the renal vein is flabby and typically collapsed. We used to have specimens that were injected. Did they inject the specimens that we're using too? Just the red and blue? Oh, they did do yellow in the collecting system. So red's in the arterial system, blues in the venous system, and yellows in the collecting system. And there's only 4 parts to the collecting system: minor calyx, major calyx, pelvis, and ureter on that. Those are easy ones to identify. Our notes here state the renal pelvis is a funnel-shaped tube that is designed to collect urine coming from our calyces. Minor calyx or calyces, cup-like structures enclosing or surrounding the tip of a pyramid, which is called the papilla. And we did our diagram above, see above. Also, see above. I'm going to take out some of these spaces so we don't have so much page stuff to actually go through. Nerve and blood supply. Interestingly, our kidneys receive about 25% of our cardiac output at any given time, those two little beans there. They're actually pretty darn small, but yet getting a quarter of our cardiac output. The nerve supply is called the renal plexus, and you've heard the term plexus before, and that's been always associated with our nervous system. And for us, it's sympathetic. Remember, it's blood vessel diameter we're controlling, and BV's blood vessel diameter is controlled by the sympathetic nervous system. And where you'll get your best control of flow through the kidney is when you control the arterial diameter. Remember, arterioles are right before capillary beds. If you squeeze them on down, you'll get less blood flow through a capillary bed, which means that everything else backs up and you don't need as much blood flowing through the entire organ itself. So that's an important thing to actually realize. Arterial diameter is the area for best control of blood flow. That renal artery is nice, but it's not going to actually be able to do as good of a job with the when we actually control the arteriole. So now here is the nephron, which is the reason why we use nephric or perinephric a lot in our discussions of the kidney. Nephron is super, super important. It is the structural and functional unit of the kidney. We have about 1 million nephrons per kidney. So 1 million nephrons per kidney, which are a very important unit for creating filtrate. Here is actually a great picture that gives you everything that you should be able to actually identify off of your kidneys in your kidney dissection work. So this is a great thing. I suggest you review it before you actually do your lab this week, if you haven't already. Couple of things that we need to do when we talk about the nephron. We need to understand what makes it up. It's a tubular system, and it has a very interesting vascular anatomy to this tubular system. And so we have to talk about the vascular aspects as well as the tube itself. The first thing that you will encounter when we actually start talking about the nephron is that thing called a renal corpuscle. Corpuscle is a typical term that we utilized previously in the past with like a blood vessels. White blood cell used to be called white blood corpuscle. Red blood cell used to be called a red blood corpuscle, but then they shortened it down to a cell. And now we use this term corpuscle for the beginning of our nephron, and I've never quite understood why they've did that, but that is what it is. And realize when you see corpuscle, it's kind of a long word. It's a two-structure unit. We have what's called the glomerulus, which is the vascular aspect of the corpuscle, and then we have the capsule, which is the tubular aspect of the corpuscle. So the glomerulus is vascular and the capsule is tubular or collecting aspect. And it has it broken down here into a visceral layer and a parietal layer. So I want you to realize parietal, remember, means lines a cavity, and visceral means it's on the structure inside the cavity. Well, the structure inside the cavity is the glomerulus. It's this weird capillary network. And so we have a visceral layer on this weird capillary network, and that's what that's referring to. And then the parietal layer is actually what's making up this capsule around it. So that's going to let you realize that then there is a space between the two, and that's where filtrate first gets formed. So that's the area of first formation of filtrate, which is of our blood itself. Then, after that, we're into more of a recognizable tubular area. So we have the first part, which is called the PCT, or proximal convoluted tubule. So convoluted, it's squirrely whirly all over the place. And when you take a look at the picture here, it should kind of look like that. and then we come down to the next part of this tubular system called the nephron loop, which is in a different area than the proximal convoluted tubule. Realize the PCT is in the cortex and the nephron loop is in the medullary pyramids. It's a very important distinction of their location. Then the tubule gets a little bit thicker again, and also kind of squirrely-whirly all over the place, and that's our DCT, or our distal convoluted tubule. The lumens look very different. The lumens, remember, are the inside, and how the filtrate is, the composition of the filtrate will be also very different when we're in the luminal side of all of this tubular aspect, and we'll learn more about that. The collecting duct, CD collecting duct, if you'll notice, is also in the cortex and goes through the medullary pyramids to where it finally terminates at the papilla, or the apex of our pyramid, and that's where urine actually comes out of. Urine comes out of the final area, terminus of our collecting duct. These blood vessels would be our arcuate blood vessels because they're going over the base of the pyramid. Remember, the pyramids are upside down, and so that would be the base, and then these guys would be our cortical radiating blood vessels, because that's what they are. They're radiating off of the arches into the cortex and giving rise to the two very important arteries going to the glomerulus. And we have an afferent arteriole, which goes into, and we have an efferent arteriole, which goes out. Now, is this normal for an arteriole to give rise to capillary bed, and then that capillary bed gives rise to an arteriole? Is that normal? No. So this is why kidneys are so unique. They have an arteriole giving rise to a capillary bed giving rise to another arteriole. And I want you to take a look at the diameter of these arterioles in this cartoon, because the diameter is designed to be distinctly different. One is larger than the other, and the afferent arteriole is larger than the efferent arteriole. And so this has a big implication for filtration pressure. You guys remember capillary dynamics and how much you loved capillary dynamics? This diameter differential means that inside this capillary bed we have what? High or low hydrostatic pressure? High. So we have a big hose leading into a capillary bed that has a small hose leading out. So that means high hydrostatic pressure, which means formation of filtrate. It is designed to favor the formation of filtrate. And so this is very important. The other thing, too, is the efferent arteriole will eventually give rise to another capillary bed, and two different types of capillary beds that will then also end up with a venule on the other side. So this is a complex arterial network that we have to realize is what contributes to the functionality of our nephrons. So back to the written part of the notes. Renal corpuscle, glomerulus, which is a very modified capillary network, plus the glomerular capsule, which has, remember, the parietal layer and the visceral layer. So we have a space to collect filtrate, which is first formed there. Renal tubule, proximal convoluted tubule. We will find in there histologically cuboidal cells with microvilli. Why do we have microvilli? Absorption. Increases surface area. So remember, whenever you see microvilli, you know there's an absorptive feature going on in here. And so this is very important. This microvilli helps this proximal convoluted tubule to be the main area for reabsorption of solutes. And it's a really weird process, because we're going to see as we go through the notes, your kidneys, those little nephrons, 1 million per kidney, actually will cause a formation of 180 liters of filtrate a day. and it's in our notes, we'll get to it, but 180 liters of filtrate a day. And nobody pees out that much urine. So we have to absorb over 99% of that filtrate back into our system. So it's a really weird process, but it works really well because everybody knows when somebody loses a kidney or two, they're in big trouble. So this process is very effective in actually getting rid of toxins and bad things out of your system, and also helping to preserve our fluid and electrolyte balance and hydration status. So it's really kind of strange, but works really well. So loop of Henle is the old name. We're trying to switch to nephron loop, and that is going to be mostly simple squamous epithelium. So that tells you this area is not an area where reabsorption is high priority. We have to let you know that different things happen in the descending limb and ascending limb. And so that's why we have them listed differently, because different things happen. But we know our loop of Henle and descending and therefore ascending limb are all located in the pyramids, right? Then we get back to the next part of the tubule, and that's the distal convoluted tubule, and we have cuboidal cells without microvilli. We will still get some absorption in this area, but that's not the main feature of this area, especially since we have so much microvilli in our PCT. And so we'll call the proximal convoluted tubule PCT, and we'll call the distal convoluted tubule DCT because it's so much easier. Way off to my side here, I have two different histology views of a glomerulus and therefore its corpuscle and the tubes and all that type of stuff. So take a look at the parietal layer of the capsule, and how would you describe those cells just by looking at the histology there? Simple squamous epithelium. Okay. This here is our glomerulus, and so it's a squiggly wiggly, squished up, mushed up capillary bed that has some very important cells covering them up, creating that parietal, visceral layer. But this is where our first filtrate comes. So it's hard to write on this darn thing. First filtrate is in that space. Taking a look here, we have one where you can see the space all the way around the glomerulus, and the other where you see this area that's called a pole. That's a pole that actually has a very special name. It's called the juxtaglomerular apparatus or juxtaglomerular complex. So that pole is where the afferent and efferent arterioles are actually located, and there's also a distal convoluted tubule there that's important to realize is there, that's actually serving as a chemoreceptor site. We'll get into that in just a moment. Based upon our previous definitions of DCT and PCT, distal convoluted tubule and proximal convoluted tubule, one of those will have a lumen that will not be fuzzy. Which one will have a lumen that will not be fuzzy in its appearance? DCT. And I say that to you because now you can take a look at the histology on these two different stains here, and you can figure out now, yes, which one is a DCT and which one is a PCT, based upon me just telling you that, correct? This one here, what is that? This guy here. That's a DCT, okay? And then here's another one to figure out. That's probably a DCT. This one's probably a DCT. So now, when I say that, let's take a look at this guy here, and he looks like a PCT. The other thing too, now you know how to look at the lumen to actually figure out, that's the first place to start. Don't you notice that the cytoplasm of those cells are kind of different in their uptake of the stain? So they do have a couple of features that make it relatively easy to figure them out. You can use the same process down below at this. And so what would you call that guy? DCT. Here's another one that looks like a DCT. Here's another one that looks like a DCT, because they are definitely different in their stain uptake, but the lumen is easy to actually view in those. And then when I erase that, my marks, oops, so much for erasing. When I erase and then start to circle these guys, I think it makes it clear to figure out those must be PCTs, proximal convoluted tubules. So that's how you approach histology. You try and take a look at them and say, these guys all look similar, and then these guys all look similar. So what is it that are different between the two? And the luminal contents, or the appearance of the lumen, and therefore even the stain uptake of the cells. It really does help. Going down to the renal corpuscle, glomerulus plus glomerular capsule, parietal layer, simple squamous epithelium. You guys very nicely identify that. Visceral layer. Interesting, we call those cells podocytes. Podiatrists, foot doctors, podocytes. These are cells that have feet. And these feet are going to form filtration slits around the glomerular capillaries. And so what you have to realize is these glomerular capillaries are under high hydrostatic pressure. And capillaries are simple squamous epithelium, correct? Or endothelium. And we can have tight junctions, we can have fenestrated ones, and we can have sinusoidal capillaries. And we want, this is a system that's designed to produce a lot of filtrate, so a lot of leak. And with these guys, these capillaries are actually fenestrated. Fenestrated little windows and are leakier, allowing us to make more filtrate. Well, if you're more leakier, are you stronger or less stronger? You're less stronger, and the podocytes are absolutely vital to the integrity of our glomeruli, because they act like say you have a hose that's suddenly spraying all over the place, and you grab the hose and you go ahead and you try to interlace your fingers and everything as best as you can, or not, but you're trying to actually stop everything from spraying out and disrupting the hose even further. And so that's what the podocytes do. The podocytes are there to actually reinforce the integrity of these very delicate capillaries that are under high hydrostatic pressure. And so the podocytes are a very important structural protective measure for our glomeruli. And our little picture down here shows you these guys here with the nucleus in this area, and their little digits, so to speak, that are going off, and they're actually interlacing with other digits of the podocytes, trying to create the filtration slits, because we do need filtration slits so that the fluid gets out, because that's the function. If I'm making 180 liters a day with these guys, fluid is getting out, and we call that fluid filtrate. Again, the fenestrated capillaries, and this is specifically designed to allow the passage of solute-rich fluid into the tubule. And I'm going to say capsule, because that still is the beginning of this tubular system. It's a collecting area. Blood supply to the nephron. Afferent arteriole brings blood to the nephron, larger diameter. Efferent arteriole, smaller diameter, and then, because the word efferent takes blood away from our nephron. But it's still arterial blood. So that means that we did not take any oxygen out of this, because that's not what we're working on. We're not working on taking oxygen out of the blood. We're working on taking out things that can actually hurt you. And we have to decide what's good and what's not good. And that's part of what the whole tubular system is all about. There are two other capillary networks that are true capillary networks: arterial capillary bed, venule side. So that's what we have here with our peritubular capillaries and our vasa recta. Peritubular sounds like it's around the tubes, and indeed they surround the PCT, DCT of the tubes. And their function is to reabsorb, which means take the stuff that's in the filtrate that we want to keep in our body back into the blood. And if we reabsorb solutes, if we bring our solutes back into our system, our vascular system, what always comes with solutes? Water. So this system that we've learned in the past with osmosis is still playing a big role here in the kidneys. So we reabsorb our solutes back into the blood via the peritubular capillaries. So that's one reason why we don't dry out immediately after one pass through our nephrons, because these peritubular capillaries are actually bringing back the bulk of what was actually created in the filtrate into our system. Now the vasa recta are going to parallel the loop of Henle or our nephron loops, and they are also going to reabsorb stuff into them to help maintain the concentration gradients of the pyramids. Because that's where we find our loop of Henle. The juxtaglomerular apparatus or complex is where we, going back up to that histology view here, this is it, that area right there. It's where the distal convoluted, a piece of the distal convoluted tubule comes into contact with a piece of the afferent arteriole right there at the glomerular capsule. Granular cells are part of the afferent arteriole, and macula densa cells are part of the distal convoluted tubule. So you have to memorize granular cells, afferent arteriole, macula densa, distal convoluted tubule. Maybe the D for macula densa will help you to remember the D for distal convoluted tubule. What's important is the afferent arteriole is feeding into our glomerulus. It can get stretched. When stretched, this will activate the mechanoreceptors, and cause contraction. So when stretched, that means the lumen's getting bigger, the diameter of the blood vessel's getting bigger, and the granular cells will cause this to be felt, and we'll actually get a contraction because we want to actually keep that afferent arterial diameter fairly constant as long as we're not under a life-threatening situation. So that's what the granular cells of the afferent arteriole will do if for some reason we get this big blood flow coming on in. The other thing is when we activate our sympathetics. They will also cause the mechanoreceptors to contract, just like we learned previously. Increased sympathetic stimulation will actually cause vasoconstriction. So this is but it will also cause, when the sympathetics stimulate, they will also cause the granular cells to release granules of renin. Renin is a potent vasoconstrictor. So two things will stimulate the mechanoreceptors. Stretch causes them to actually contract on down, or sympathetic nervous system causes them to vasoconstrict and release renin for further vasoconstriction. Macula densa cells in the distal convoluted tubule. These are chemoreceptors. And they detect sodium chloride levels. And if the salt of the urine is too high or too low, it will also adjust the flow of blood and therefore filtrate production in our glomeruli collecting tubule system. So all I want you to know is, I don't want you to know which way things go, I just want you to know they're chemoreceptors and they measure sodium chloride levels to adjust formation of filtrate. And I hope that you can realize when you increase flow to the kidney, you will increase the formation of filtrate. When you decrease blood flow to the kidney, you will decrease the formation of filtrate. So blood flow is very important in formation of filtrate. Collecting tubules receive filtrate from many nephron units, more specifically the distal convoluted tubules of each of the nephron units, and will empty into tip of pyramid, which you know is the papilla, and then urine into minor calyx. And now we're into the beginning of kidney physiology, or the production of the filtrate. So whenever I say filtrate, you have to realize that means we're in the tubule system. We're either in the capsule, capsular space, the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, or even the collecting duct. Filtrate is always in that. Once it leaves the collecting system via the apex of the pyramid, that is actually urine. And its concentration has been determined before it actually exits the collecting system here. We have three different colorations here. Glomerular filtration is what we're going to talk about first. Then after we go through that mechanism, we'll talk about tubular reabsorption, which means we want things back into our bloodstream. We've made 180 liters and we want 179 liters back into us almost. And then tubular secretion. That is when we actually have bad things in our system. Our kidneys are actually tools that can help us if we are acidotic. If we are acidotic, what do you know about pH? Low. What do you know about protons? High. Okay? So what's really important is one of the mechanisms that we have to compensate for an acidosis, and let's talk about a metabolic acidosis because that's the most common form of acidosis, is creating a respiratory alkalosis, correct? And so to do that, we have to hyperventilate. Well, if that doesn't work, then you have another buddy that will help you out with the acidosis, and that is the kidneys. And so if you have too many protons floating around in your bloodstream, your kidneys will help you out via tubular secretion by actually forcing protons into the filtrate, which means it acidifies your urine, correct? And so we'll have to have a nice buffer system in there to help out with the acidified urine, otherwise it burns when you pee. So starting with glomerular filtration in our capsule. Glomerular filtration is passive, which means it doesn't require ATP, and it is non-selective, which means it takes out everything. That is not cellular. Okay, so it doesn't take out red blood cells, doesn't take out white blood cells, and doesn't take out platelets, but takes out everything that you send its way. That's what non-selective means. the solutes, we'll take all comers. So it allows the passage of all except for proteins, basically large proteins like albumin, and our blood cells, red blood cells, white blood cells, and platelets. And so in the formation of the filtrate, that is going to result from net filtration pressure, which is GHP. G stands for glomerular hydrostatic pressure. And then we have a glomerular osmotic pressure, which counteracts hydrostatic. I hope you guys remember. Hydrostatic pushes the fluid out, osmotic pulls the fluid in. And then we also have this C, but our C here isn't for capillary, it's for capsular hydrostatic pressure. And glomerular hydrostatic pressure is pushing the fluid out of the glomerular capillaries into the corpuscle, or the capsular space. And as that capsular space fills up with fluid, it's a back pressure. Can you guys follow this? We have hydrostatic pressure and we have back pressure. When you fill everything up, can you push more fluid in if you've got it completely full? No. So that's what capsular hydrostatic pressure is all about. It's telling you that there's some fluid in the capsule, and that creates a back pressure. And so net filtration pressure is glomerular hydrostatic pressure minus glomerular osmotic pressure plus capsular hydrostatic pressure. And we don't actually worry about that capsular osmotic pressure because it's actually, you'll see later on, it's a number that's doesn't actually enter into this calculation. We have some numbers here. Net filtration pressure is going to be glomerular hydrostatic pressure of 60. 60 is pretty darn high, isn't it? In a capillary bed, what was our arterial capillary pressure in the beginning of our normal capillary beds? 35. So we've gone from 35 all the way up to 60, and we're also fenestrated. So those podocytes are very important. Now we have a glomerular osmotic pressure of 32, and what I'm going to tell you is I'm going to round these numbers to round numbers. You guys have a problem with math. I'm just teasing you. But let's keep the numbers round. You know, it makes it a lot easier. So we'll take that to be 30. So what do you suppose this one is going to be? 20. Absolutely. So this number here, 30 and 20. And I just do this for convenience. These are easier to remember, I think, than 32 and 18. Maybe now you'll always remember 32 and 18 because I've done this. But the osmotic pressure of the glomerulus is 30, and the back pressure of the capsule is 20. So our filtrate is formed with only a net filtration pressure of 10 millimeters of mercury. So that's where we're going to stop today. We're going to pick up with this topic again next time, and we're going to talk about that's all we need on a daily basis to make that 180 liters a day of filtrate. So what happens when we actually monkey with these numbers? And that's what we're going to talk about next time.